Question 9. Question 9. The diagram below shows how a human gene can be inserted into bacteria to produce human insulin using recombinant DNA technology. Nice little picture of that. Yet again, it's a very clear diagram of genetic engineering. If you wanted to lift that and just add to it, it would be a nice one for a mind map. Okay, name one enzyme used in this process, state its function. Okay, we have two enzymes. You've got restriction endonucleases. And if you name that one, then what we're looking for is that cuts out the gene, or cuts open the plasmid. Okay, and if you wanted to be posh about it, but it's not in the mark scheme, but you do need to know, remember that gives you complementary sticky ends which will allow them to seal. Okay, which brings you to the second enzyme, which is ligase. So ligase joins DNA. Let me be more precise about that. Joins DNA. So that's the gene to plasmid. Okay. The recombinant plasmid also contains a gene for resistance to the antibiotic ampicillin. Suggest a procedure which would allow only cells containing the recombinant plasmid to be selected. Okay, well, this is pretty straightforward. So you add ampicillin to the culture, and only bacteria with the plasmid will survive. That's it. So you've got two parts to this one. First one is adding the ampicillin, and the second one is saying that would mean that only the recombinant ones will survive. Okay, plasmids with these antibiotic resistance genes have been passed to other bacterial species by horizontal transfer. Describe the process of horizontal transfer. So horizontal transfer is the transfer of DNA between individuals of the same generation. I think that's the simplest one that you've got. You could properly explain it and describe the movement of information from, say, one bacteria, copying information down through the modified pilus to another one. That's via conjugation, so you could explain that process. Or you could do transformation in terms of just taking up small sections of DNA from the culture around about it. Or you could have movement by viral vector. You could describe those very clearly and that will get you the mark, or you can just define horizontal transfer, meaning that it's transferred between individuals, same generation, not passed on through sexual or asexual reproduction to the next generation. Okay. When culturing the bacteria which produce insulin, sterile conditions are maintained. Explain why this is important. So basically this removes competition, which means you only get desired bacteria. As well as removing competition, you could put that as the same way you could also say it removes contamination. So that means that you wouldn't get, again, the things you don't want being produced. And that's the question.